Hello everyone who watched this video. In this video, we will be reviewing a paper called A Global Test of Hybrid Ancestry from Genome-Scale Data. First of all, let us introduce ourselves. My name is Annette Jasmine and I'm with...
Aisyah Annette, hello.
Hello! Okay, let's moving on. So what is the problem from this paper? Well, it is about a new statistical method for detecting the presence of hybrid species within a group of species using genome-wide DNA data. Why and how we do it in evolutionary biology, there is also a chance the probability that a species can be a result of hybridization. Relationship between species are generally believed to stem from a common ancestor, but there is a possibility to it. And well, the research gap from this paper is that the older methods only test a single species at a time as a hybrid candidate, but in this paper they tested in a global analysis. And that also them overlook many incomplete lineage sorting, which is the possibility that the trees from different species are maybe mistaken for being hybrids. Well, the data that I use are covering some evolutionary scenarios, which is where when the hybridization happens, there are three types of it. Maybe it's recently, maybe it happened a long time ago, and maybe it's recent and it happened a long time ago, or it is recent and it's in many species to be with, and then this is where the hybridization happens a long time ago in many species. And this paper used two types of data, simulation for testing and empirical data for validations. In the simulations, they use five species network scenarios with varying parameters, 100 gene trees per scenario for a total of 88 networks. While the validation they're using with rattlesnake dataset, nuclear loci dataset versus one mitochondrial locus with a total of 8,466 sites. And the last yeast dataset. The statistical methods that are used is hill statistic, which is to test that is there any species that is hybrid? What three methods following? The first one is to test the individual test per quartet and review it for all quartet combinations that are possible from these species that exist, and then they obtain the p-values, and then because it is related, the tests are correlated with one another, so the features test can be used, and then they use the Cauchy combination test. They then combine the CCT with the min p-test, which is sensitive to the smallest p-value.
So what are the key results of this research? Number one is developing and applying the Cauchy combination test and MINP methods in large genome data, as what my friend spoke before. And then the second one is using the new method to resolve incomplete lineage sorting, or ILS. And then the third one is trying the new method by sampling it on real or empirical data with different species like yeast and citrus rattlesnake. Next. So did the model perform well? Yes, both models perform well in detecting hybridization and distinguishing it from incomplete lineage sorting, or ILS, across different hybridization levels like different DNA single lengths, different effective population sizes, and different branch lengths. The main difference was that the MCM approach showed slightly higher statistical power than the CCT method in both simulation and empirical analysis. Next. Okay, so the main genes or biological mechanisms that are important were, number one, of course, the genes with incomplete lineage sortings, or ILS, because there are like some type of noise in the data. And then the second one is genes with hybridization. This one is the one that we want to highlight the most, the one where we want to search in different types of the genomes. And then the third one is effective population size, because the bigger population size, the better the model can catch the data, because when it is shown that the theta is 0.001, it shows a very small accuracy, but when it gets bigger, it's better. Next. Okay, so what were the key results? The key results were it detected hybridization well across three different species with different hybridization. The one that my friend highlighted before, like recent and ancient hybridations, and etc. And then the second one are their methodological limitations. Yes, the author assumes that the data pairs follow a normal bivariative distribution, whilst MCMs perform better. It is prone to type 1 errors in hypothesis testing. Moreover, the number of individual tests required becomes very large as the number of taxa increases. Next. So could another statistical approach be better? Yes, another statistical approach could be better by performing better in maintaining MCI's high power while providing better control of type 1 error. For example, like an additive weighting framework, or use of empirical null distributions through permutations method may increase the accuracy. Next.
What to improve? Well, there's three things that we think can be improved from this paper is to do tests on many more species with more than 20 taxa, because in the real world, more than phylogenetic studies can involve hundreds of species. And the second one is more empirical data sets, especially plants and locals, because hybridization is actually very common among flowering plants, and it is estimated that more than half of a plant species have a history of hybridization. The third one is tests on missing data and complete sequence, because this simulation assumes that the DNA data is perfect and complete, while however, in reality, many genes have not been successfully sequenced for all species. Further investigation is needed. How much missing data can be tolerated before statistical power drops significantly? Thank you. That is all from our review on this paper. I guess this is the end of the video. Thank you for watching.
Bye.